Today we'll be looking at excitable tissues. Can anybody guess what excitable tissues are?
No, don't do like nerve tissue.
Anything else that can be excited or triggered to do something? Cardiac, muscle tissues in general, nerve tissues, muscle tissue, they're excitable tissue. So today we'll be primarily looking at action potential in these tissues. We'll start by looking at nerve as in nerve tissue being excitable. But in order to look at these tissues, we'll still have to understand the genesis of the excitation of these tissues. The brain has many areas, and you guys already know that the brain is divided in lobes, different region, and different region of the brain have different function. What you would primarily need to pay attention to here is primary motor cortex. And you already know that motor has to do with muscle function. So you can imagine that the primary motor cortex will contain neurons, axons that are going to be running through tracts in the spinal cord to muscles in the body. The somatosensory cortex or sensory cortex is found around here. There are different names of these regions just to orient you as to where what is coming from. You don't have to know primary motor cortex or sensory cortex. As long as you know that it's coming from a specific area in the brain, that's important. I mean, there's also sensory primary visual cortex. This is where the impulses for you to see actually goes. They run in tracks and all the way to the back of the skull. You know that sensory is basically sensory perception. So what are sensory perception? You touch, you feel. Whatever you feel, you can feel pain, you can feel temperature, you can feel cold, you can feel pressure. All of these are sensory perception. Most of these nerve endings are nerve endings for pain, cold, pressure, touch. All of these are usually located on the skin in nerve endings. So for example, if you look at the diagram, you have Ruffini corpuscle. This is a touch receptor. If somebody touches you, this is where the information will go. This corpuscle here. If somebody pushes you or there's some pressure, you put somebody in your lap, whatever it is, as long as there's pressure on the skin, the Pacinian corpuscle will receive that information and send that information directly to the brain. And this is mediated by action potential. For pain, temperature, it's the same thing. Free nerve endings in the skin will receive the information from the external environment in the form of stimuli and transmit this information to the brain. So if you realize all of these sensory perception or reception are actually going towards the brain. So you'll realize eventually that they call them ascending. So they run in the ascending tract. So anything that is sensory, you're picking up the information from the external environment and you're sending that impulse to the brain for the brain to respond, which would be feel. So you would feel cold, you would feel pressure, you would feel pain. All of those are running in ascending tracts. For the motor, you'll find that it's different. Remember that motor functions, they're voluntary. So these activities are being organized from your brain directly. And before you move, your brain already processed that you will move. All of the muscular functions or muscular activities will leave the brain in what we call descending tracts. So the difference between motor and sensory is that ascending tracts for the sensory and the descending tracts for the motor function. If you look at this diagram on the right, this is just showing motor neuron here coming from the brain, and you know that it goes through the spinal cord. Remember we said that the brain, the external or peripheral region of the brain, you have the gray matter on the inside, you have the white matter. And remember that the gray matter are just cell bodies, glial cells, and it looks gray because of the rough endoplasmic reticulum. And the white matter are because of the myelinated axons and tracts that are running through. Eventually, you know that when they get to the spinal cord, there's a switch where the gray area is now in the central region. So this is where you'll have the cell bodies in the spinal cord, and the outer white areas where you'll have the axons running in tracts. From the anterior ventral region, you have motor fibers. Remember, these motor fibers should be coming from the motor area in the brain, right? It's just that the tracts, they change position or location or even switch path, but they're still continuous tracts that have specific destinations. In terms of the sensory, if you remember the first diagram, sensory should be coming from the sensory cortex. They should also be coming through the spinal cord here, and they tend to come through, some of them through the posterior area or the dorsal root. So you have the dorsal and the ventral root. So this is also showing sensory receptors in the skin, the mycinous corpuscle, Ruffini corpuscle, Pacinian corpuscle, Merkel's disc. All of these are responding to pain, temperature, touch, and this is basically just showing sensory pathways. The stimuli comes from the external environment, wherever it could be you throwing hot water on somebody. It will basically stimulate the cells under the skin, and this sends the message through sensory neuron that will go to the brain, and it will be interpreted as whatever it is. This is a more complicated diagram. It's basically just showing the tracks. So when you look at a spinal cord, if you've seen a spinal cord on histology, you guys should see one, right? I'm sure there was one. Anybody saw it?
Yes, miss.
Great. What did you see? Did you see the ependymal cells?
Yeah, I did. I saw them lining the
Ventral
the
canal. Yes,
Yeah.
very good. That's more of what you would probably see in that histology slide. You should be able to identify the gray area as well as the white area.
Yes, miss.
Yeah, but in terms of the tracks, you wouldn't be able to see them because, as you know, they're on the periphery and they're supposed to be white. But to the left, these are motor or descending tracks. Remember, we said that the motor are the tracks that are coming from the primary motor cortex in the brain, and we call them descending because of the direction of the information. So it's descending. There are different types of tracks. You don't have to know the tracks, just to orient you as to the complexity of it. You have pyramidal tracks, extrapyramidal tracks. Extrapyramidal tracks include corticospinal and lateral and anterior corticospinal, depending on where they are situated. But what's important is that these tracks, corticospinal tracks, will contain axons that will eventually lead to peripheral nerves that will contact muscles. And remember that you are actively using your muscles all the time. In order to stand, you need to extend the muscle. So if you're sitting, it's flexion. If you're, for example, working out your arm, you're doing a lot of extension and flexion. You can imagine that these tracks are very important in such actions. So extrapyramidal tracks that are also very much important in extension, flexion, they have highly specific functions in the body. Cerebrospinal tract, reticulospinal tract, olivospinal tract, vestibulospinal tract. These are all very important tracts that are for motor function. And as we said, the sensory are the ascending because they're receiving information from the external environment and they're sending it to your brain. You have specific areas of the spinal cord, again, that is responsible for proprioception. If you're standing, in order to be oriented in the environment or have spatial orientation, you need to have sensory perception. Fasciculus gracilis cuneatus, these are tracts that are responsible for proprioception, vibration sense. So if you feel vibration, it's a sensory perception and is as a result of these sensory tracts. You also have different types of sensory tracts, as the diagram is showing. The ones in red are the motor tracts. The ones in blue are the sensory tract. This is just giving a little bit more example as to the function of these tracts. For example, the descending tracts, the lateral corticospinal is the tract for voluntary motor function in the upper limbs, so the upper extremities. Ventral corticospinal tract also is very important for voluntary muscles. In terms of the sensory, this is just showing what I've said before. Fasciculus gracilis, fasciculus cuneatus, these are some tracts or axons that are running in the posterior part of the spinal cord. They are responsible for proprioception. In order to stand and be oriented in the environment, it's called proprioception. In order to move, you have to have an idea as to where you're oriented in space, and these tracts are very important for that. In terms of fine touch, small sensation, the fasciculus gracilis cuneatus, I think, is very important for fine touch. This is just showing a little bit more in terms of where they are running. So this is just the primary motor area, and you can see the tracks running through different areas of the brain, but there are different cells that are there. These cells will give extension to axons that are coming from the primary motor area, and you'll eventually realize that they give rise to the corticospinal tract. And this is just showing where the corticospinal tract runs from the primary motor area in the brain through the spinal cord, through the ventral column or the ventral region of the spinal cord, until it goes into supply muscles of, for example, the upper limbs or the lower limbs. This is just showing the pathway of sensory tract. This is the primary sensory cortex. It's showing where the fibers or the tracts run, and they run in very highly specific areas of the brain. So for example, if you have a stroke or a gunshot injury or a wound, whatever it is, if you have a damage and for example, this part is affected, then you'll have the manifestation in the limb, because once you cut off the signal or the impulse from here all the way down, then you're not going to have the response in the end organ. If you have a stroke somewhere around here and there was bleeding and there's ischemia to this region where the tracts, for example, the motor tracts are running, then this side, for example, of the body, and if you look, sometimes they call it contralateral or ipsilateral, because even though the tract starts on the right side of the body, you realize that it ends on the left side of the body. So sometimes if you have, for example, a right-sided damage to this region, then the left side of the body will be impaired. In terms of the sensory though, the sensory is coming from the primary somatosensory cortex, and it's also running through a specific area of the thalamus, the ventral posterior part of the thalamus, and if you remember, they run through the posterior area of the spinal cord eventually as fasciculus gracilis, fasciculus cuneatus, and they give rise to proprioception, vibration. There are different sensory perception. I'm just giving you guys example of the most common type, the sensory and the motor. And this is also showing the tracts, the fasciculus gracilis, fasciculus cuneatus, running from the somatosensory region. And you should imagine the same. When you have time, you can look through to read what these are saying. It's basically the same tracts that I showed you in the first picture, just showing the location of the tract. We'll have to go back to the basic, the cell membrane. There were a lot of things that we discussed about the cell membrane, but there were also things that we didn't talk about. We talked about the simple diffusion, facilitated diffusion, yes, but we didn't focus on it so much. Not that we need to focus on it. Primary active transport. What is important about the cell membrane though that we didn't focus on was the fact that you have ion channels. These ion channels are very important in terms of cell excitation. It's also important to note that the intracellular environment and the extracellular environment is going to be different. In order for excitation or excitability of any cell, there has to be the potential for electrical activity or mechanical activity. If you look at the external environment of the cell, you'll realize that there's a balance of ion in the extracellular space as well as the intracellular space. So if you look at the composition of, for example, sodium, potassium, chloride, calcium, it's going to be different in the extracellular space versus the intracellular space. So if you look at sodium in the extracellular space, you'll realize that sodium is high in the extracellular space. The concentration of sodium tends to be very high in the extracellular space, but the concentration of potassium tends to be low in the extracellular space. So just remember these two things. Yes, chloride's also high in the extracellular space, higher. Sodium, chloride, calcium tend to be on average somewhat the same, but for sodium, chloride, remember that they're much higher in the extracellular space, while potassium tends to be lower in the extracellular space but higher in the intracellular space. So if you look inside of the cell, you realize that the concentration of sodium is 15 millimoles, potassium is 120. If you look at the external space, you realize that potassium is 4.5 on average, 4.5 to 5. So this is a big difference. It's like potassium is 120 within the cell, but 5 outside of the cell. Potassium is 145 outside of the cell, but 15 within the cell. So these are all important information because they will determine the membrane potential, or they will determine the progress of action potential. So what's important about the cell membrane though, because we're still looking at the cell membrane here, this is supposed to be, you might not be seeing the phospholipid layer, the hydrophobic, hydrophilic region. Just imagine that it is there because it's nothing that you don't know. The important thing about the cell membrane is that it will have sodium-potassium pump. It will need sodium-potassium pump to regulate the amount of sodium and potassium that goes within and outside of the cell. So if you look at the sodium-potassium pump here, what the sodium-potassium pump will do, it will create a balance of sodium within the intracellular space and also in the extracellular space. So the pump will pump out 3 sodium ion while it exchanges that for 2 potassium ion. So this pump, 3 sodium will leave. For every 3 sodium that leave, 2 potassium comes in. You also have potassium channel that are separate from the sodium potassium pump. The potassium channel will allow potassium to leave negligibly, and you also have voltage gated channel, well as ligand gated channel. You're not seeing this in this diagram, but these are very important structures in the cell membrane that will help to regulate action potentials within the cell. So again, we said that you have different ion channels on the cell membrane. Ligand gated channel. Ligand gated means that it responds to a ligand. A neurotransmitter can be a ligand. For example, acetylcholine can bind to the acetylcholine receptor, and that would be like a ligand gated response. So when the acetylcholine goes to the neuromuscular junction at the postsynaptic terminal and it binds to the receptor, it's seen as a ligand. What it will do, it will cause an influx of sodium within the cell. Remember that when the sodium goes into the cell, it will stimulate the release of calcium from the sarcoplasmic reticulum. So this is one way in which you can stimulate an action potential via ligand gated receptor channel. You can also have mechanically gated ion channel, meaning that they respond to touch or pressure. So if, for example, the nerve endings on the skin, I touch your skin, this will stimulate a little vibration or a little pressure, and the nerve will sense that pressure by causing an influx. So when the nerve actually senses that pressure, this will automatically cause these channels to open. So you have sodium, mechanical gated sodium channels that can just open as a result of me touching you. But if, for example, the touch is not significant enough, it won't stimulate an action potential. You also have the potassium and sodium, as we said, that can be always open, and you have voltage gated. So the ion channels can be voltage gated, ligand gated, mechanically gated, are always open. And this is just showing again the intracellular versus the extracellular compartment. It's also showing the resting channel of potassium that is always open. In the neuron, remember that the potassium is higher in concentration in the intracellular compartment. The sodium is higher on the extracellular compartment. In order to stimulate an action potential, what will happen is that the voltage gated channel will have to open and a lot of sodium will have to enter. So you call that sodium influx. A lot of sodium will have to enter the cell, and this will basically cause what we call depolarization of the cell itself. And when that gets to a peak, it goes to repolarization, and that's when you have the action that is stimulated. But in terms of the no action, you have a resting membrane potential. So the cell at rest has a resting membrane potential. The resting membrane potential means that there is a balance between potassium on the inside of the cell, because the inside of the cell will be negatively charged. So you'll have a certain amount of potassium on the inside of the cell, and then you also have a balance of sodium in the extracellular space. When the cell is at rest, there's a specific number of potassium that is going to be on the inside of the cell, and there's going to be a specific number of potassium that is going to be on the outside of the cell. So this resting potassium channel is open at rest, and you have probably 1 or 2 potassium ion going out, and it stabilizes the membrane potential. At rest, there's a specific amount of potassium that's going to be in the intracellular compartment, and there's going to be a specific amount on the extracellular compartment or within the extracellular space. In terms of the action potential, remember that we said that the voltage gated channels will have to be open now when you have an active action potential, but at rest the voltage gated channel are closed. Once you have an action potential, the voltage gated channel is going to be open. So when the voltage gated channel is open, it causes an influx, a massive influx of sodium within the cell. Remember, it's not natural for a lot of sodium to be in the intracellular compartment. So that is the difference. When the sodium moves from the extracellular space to the inside of the cell as a result of some form of stimulation, it could be a stimulation from acetylcholine receptor, it could be a stimulation from me touching you. This will result in sodium rushing into the cell, and depending on the amount that rushes into the cell, it will change the cell from a resting state to an active state where it stimulates an action. So it can stimulate a contraction, it can stimulate a sensory reception of pain. So we already mentioned this before, ligand-gated channels, they're chemically gated channels, they're open when neurotransmitters bind. It doesn't have to be only neurotransmitters. Drugs, for example, interact with ligand-gated channels. Hormones interact with ligand-gated channels. You find ligand-gated channel as well as voltage-gated channels throughout the nerve cells. So you find them on the dendrites, you find them on the cell bodies, you find them at the axon hillocks. I remember mentioning that at the initial segment of the axon, you have a lot of channels between the nodes of Ranvier. Remember that we said that the oligodendrocytes or the Schwann cell myelinate segments of the axon. So the segments that are not myelinated, you call them the nodes of Ranvier. It's like the gaps between the myelination. What you will have are channels, a lot of ligand-gated channels, as well as even voltage-gated channels. And these are going to be important in what we call a saltatory conduction. So when you have an active action potential and you want the polarization of the neurons or the cells, the ligand-gated channel will help to reinitiate action potential along the nodes of Ranvier. I already touched on the mechanically gated channel. We said that they're just there. They are not, in general, they don't stimulate rapid action potential. They're just there to stimulate the cell so that it takes it, for example, to a sub-threshold state. And you'll eventually understand that in order for an action potential to take place, the cell has to be at rest first, and then you have a stimulation. When the stimulation comes, the stimulation can be sub-threshold. If it's sub-threshold, it's not going to stimulate the active action potential. Once you get it above the sub-threshold to the threshold, that's when you get the action potential. In terms of excitation and conduction of the nerve cell respond to electrical, mechanical, chemical signal. Electrical, we know that with the inside of the cell, it's more negatively charged, which is why you're seeing, if you look at this graph, you're seeing a resting membrane potential. Nerve cells tend to have a resting membrane potential around minus 70 millivolts. The cell is at rest, nothing is stimulating it to do anything, it's at rest. So it's around negative 70 millivolts. You can can have stimulation that or a generator potential. This is where we say it's sub-threshold. Once it's within this area, for example, if I touch you and the mechanical ion channel stimulate an influx of sodium, the sodium is not enough to take it to a threshold. So once the stimulation is enough to take it above the sub-threshold, once it gets to the threshold, that's when you have the rapid influx of sodium, and that's when you have the depolarization. So if you look at the graph here, you see depolarization. This is basically when the voltage gated sodium channels are open, and when it gets to this area, that's when the action potential is carried out. So once action potential is carried out, you don't need the sodium or rushing at the cell no more. So what the sodium channels, voltage gated sodium channels will do, they will eventually become inactive or deactivate. So that will cause a closure of the sodium voltage gated channel and repolarization. This is when the potassium now starts leaving the cell, because you need a balance. Remember, you already have a lot of potassium within the cell, and then you're going to bring in a heap of sodium to create this chaos. The potassium will have to leave slowly in order to repolarize and get back to that resting state. And how it does that is by the sodium potassium ATPase pump. This is basically an electrical activity. So you have the negatively charged, a lot of negatively charged ions within the cells, negatively charged protein, which is why you have the negative here. If when you have an action potential, you move from a threshold to depolarization to the more positively charged end. So with an action potential, the cell leaves from a resting state to a more positively charged state, then repolarization as well as hyperpolarized state. In terms of excitation and conduction, electrical impulses are measured in milliseconds. So the potential charge measured is in millivolts. So remember, we're dealing with negative and positive charge. It's electrical activity that we're dealing with. Impulse is normally conducted along axons and along axon termination. So remember that once you get the stimulation, it goes along the axon and it jumps across the nodes of Ranvier and goes to the axon terminal. So this is just showing another image of the resting membrane potential. We said the resting membrane potential of the nerves around 70 millivolts, and the sodium gates are open. In this case, it has to be at a threshold, and the sodium gated channels are open. You have rapid influx of sodium ion, which causes a depolarization. When this depolarization occurs, naturally there has to be a limit to the amount of sodium that comes in. So after it gets to this level of the action potential, the potassium gates will open to allow the potassium to leave, to create a balance between the sodium and the potassium, because so much sodium was not supposed to be in the cell at a normal state or at a resting state. So there has to be some regulation of this. So the repolarization is when the potassium ion comes out of the cell, and then you basically have a balance between both of them. So this is just showing membrane potential. Remember, I said that membrane potential is basically a balance between the amount of sodium in the extracellular space and the amount of potassium in the intracellular space. And we know that potassium can diffuse negligibly out, but not a lot, right? That's when the cell is at rest. You have a little one or two, depending on if you need that balance. And for example, at repolarization, you have the sodium potassium pump that will push out 3 sodium and take in 2 potassium. So in terms of the resting membrane potential, separation of negative and positive charges across the membrane. If the inside of the cell is negatively charged, or negatively charged proteins, a lot of negatively charged molecules are within the cell. So inside of the cell is negatively charged. So it's a balance or a separation of negatively charged inside of the cell, which is why when you start the action potential, it moves from negative 70 up to positive 35. Positive positive charge across the cell membrane. Unequal distribution of ion, one or more ion species across the cell membrane. So this, you can have diffusion across the cell membrane where you have try to have a balance down a concentration gradient. So diffusion can occur as well. This is just saying what I mentioned before. The resting membrane potential in neuron is around 70 millivolt, and this is also just a diagram showing the action potential from the threshold to the depolarization, the opening of the sodium channel, closure of the sodium channel, opening of the potassium channel, and closure of the potassium channel, and the repolarization. In resting membrane potential, we know already that the potassium is higher on the inside, sodium is higher on the outside. The sodium potassium ATPase is what regulates the amount of sodium potassium, or creates this balance between the cell, or between the cell membrane. So this creates a resting membrane potential. So the inside of the cell is negative at rest. This is just showing a neuron myelinated and unmyelinated action potential. You know, the difference between the myelinated would be that the action potential will be more rapidly conducted in this case, right? And you have the nodes of Ranvier that will reinitiate and cause the saltatory conduction. This is just reinforcing what I've said before. You have the sodium potassium pump, where for each 3 sodium that leaves, 2 potassium enters the cell. The cell is more permeable to potassium at rest, and the resting membrane potential is dependent on potassium. So this is just showing a nerve and action potential in the nerve. You can imagine that this is the soma or cell body, and this would be the axon. Imagine that it's myelinated. So for example, an area of the nerve is stimulated, the voltage gated channel will open, and the voltage gated sodium channel will open, and will basically cause a lot of sodium to rush into the cell, and that's depolarization. So here you can see that the sodium channel is open, and this is depolarization. The sodium will diffuse in the cell, creating a change in polarity of the membrane ahead of the potential. This change in polarity activates the voltage gated channel along this part of the membrane to open, letting more sodium rush in. So initially you're gonna have the sodium, but it is when the voltage gated channel are open that you will have an action potential. So you can have sodium channels being opening, and you have sodium ion coming into the cell, but it's only when it gets to the threshold that you will have the voltage gated sodium channels that will open. Otherwise, if the voltage gated sodium channels are not open, the action potential cannot be generated. This is also showing hyperpolarization. After the action potential, the sodium channels will close and the potassium will open. So you have an efflux. So the potassium will have to leave, because so much of the sodium coming in, the potassium will now have to leave to create this balance, and this balance will eventually be stabilized by the ATPase pump. There is the equilibrium potential of sodium, where the sodium is at a balance, and that's before it gets to the threshold. Basically, you know already the action potential in a nerve will start by depolarization stimulus. So you have a stimulus for depolarization. The stimulus does not have to initiate depolarization. It can be a stimulus for depolarization, but it does not trigger depolarization. It does not trigger depolarization because it's at sub-threshold. It only triggers depolarization when it gets to the threshold. And that's just the important thing to remember for this. So once the threshold is reached, a full action potential is produced. Action potential fails to occur if the stimulus is sub-threshold in magnitude. Nerve cells are polarized at rest. Positive charge are on the outside, negative charge are on the inside. That's a lot of repetition for you. In terms of the polarity, it's briefly abolished during the action potential. And the voltage gated channels, as I said, are highly concentrated in the nodes of Ranvier and the initial segment in myelinated axon. So you tend to have a lot of sodium channel per square micrometer of the membrane in myelinated neurons, and they've been estimated to be up 50 to 70 in the cell body. This is just showing the nodes of Ranvier. So this is supposed to be the myelin sheath, and here should be the nodes of Ranvier. So where you have the cell body or the first neuron, you tend to have a lot. Where you have the initial segment, you tend to have a lot more of these sodium voltage gated channel, and not only sodium, as you can see from this diagram. This is showing the area of a node of Ranvier. You have potassium voltage gated, sodium voltage gated. Just know that you have a lot of these potassium and, well, a lot of the sodium voltage gated channel naturally to stimulate depolarization. You can have up to 2,000 to up to 12,000 at different segments. Depolarization in myelinated axon travel from one node of Ranvier to the next. Jumping of depolarization from node to node is called saltatory conduction. It is a rapid process that allows myelinated axon to conduct 50 times faster than unmyelinated. So that's a big difference. And this is just showing the sodium channel and the active node and the peak of action potential. So in terms of the muscles, skeletal and smooth muscles, they are also like neurons. They can produce or respond to chemical, electrical, mechanical stimuli or create these functions. The difference is that action is in contractile protein. Cardiac myocytes, skeletal myocytes, they are similar in structure. So even with smooth muscle contraction, you'll still have the actin binding the myosin and the myosin head, and the whole contraction is similar. We already know there are 3 types of muscle. What's important to take from this is that the cardiac muscles have pacemaker cells. So even though you know that the cardiac muscles get the sympathetic, parasympathetic supply from the autonomic nervous system, these nerve supply, they're only able to speed up or slow down the heart, but they do not generate a natural contraction. These pacemaker cells in the heart stimulate themselves spontaneously, and your heart is formed from around 5 weeks gestation. So when you're like 5 weeks, you have a heartbeat. Pacemaker cells have their own potential, so they beat spontaneously or trigger action potential spontaneously. It's just that they're assisted by the autonomic nervous system if, for example, you need to speed up the heart or slow down the heart. But if you take a person's heart out of them chest, it will still beat because the pacemaker cells, they derive their own mechanism of contraction. Smooth muscle lack striation, as you already know, they are found in the hollow viscera, and they also have pacemaker cell, but they discharge irregularly, not like the cardiac muscle. Contractile mechanism in skeletal muscle largely depend on myosin 2. Remember, the myosin has 2 heads. They also have the actin, tropomyosin, troponin, ITC. This diagram you're already familiar with. I don't have to go through all the detail with you. You know that the tropomyosin is here and the troponin is here, and you have different types of troponin. The sacrotubular system, you know, we find that in the skeletal as well as the cardiac. This is very important for the muscle excitation. This is where the net effect of the action potential is truly generated. So even though it's coming from the nerve terminal, you know now that it's coming all the way from the motor cortex in the brain. It runs through tracts, for example, corticospinal tracts that goes to peripheral nerve, a skeletal muscle. You know that once that action is generated in your mind, because you're directing all of these actions, that goes through to the nerve terminal. Acetylcholine is released, acetylcholine receptor is stimulated, sodium voltage gated channel channel opens, it basically causes an influx of the sodium into the T tubule, and then it will stimulate the calcium release from the sarcoplasmic reticulum. And remember that the calcium will bind to the troponin, and the troponin will remove itself to release the tropomyosin. It's, I think, specifically the troponin C. Calcium will bind to troponin C, and the troponin C will release the area on the tropomyosin so that the myosin, it can bind to that region and it pulls it to the midline. So in terms of the electrical characteristics of skeletal muscle, the electrical event is similar to the nerves, where the only difference is the resting membrane potential for the muscle or the skeletal muscle. It's around 90 millivolts. For the cardiac muscle, around 80 millivolts. So it's nothing that you need to commit so hard to memory. Nervous tissue around 70 millivolts, cardiac muscle around 80 millivolts, skeletal muscle around 90 millivolts, smooth muscle around 60 millivolts. The action potential in skeletal muscle lasts 2 to 4 milliseconds. Depolarization is also triggered by the influx of sodium, just like the nerve tissue. Repolarization is efflux of potassium. So this is just showing the action potential. They have the resting membrane potential. Remember, the resting membrane potential is the balance of the intracellular and the extracellular ions. So when that stimulus comes, it triggers depolarization by opening the sodium channel, and repolarization will basically cause an efflux. So it opens the potassium channel in this case, and potassium leaves the cell. This is just reinforcing the intracellular versus the extracellular compartment as it relates to muscles. The sodium concentration tend to be a little bit higher, 12. Extracellular, it's the same, 145. Potassium, 155. It's the intracellular compartment, so you expect the potassium to be higher and the potassium should be lower in the extracellular compartment. So if, for example, you're a normal patient and you come to the hospital and we take some blood from you and we do urea and electrolytes, the electrolyte would be the sodium, the potassium, the chloride. The potassium being very high in the extracellular space, for example, if it's above 6, 7.5 in the extracellular fluid, you are at serious risk of dying, because the potassium can trigger the heart to have fibrillation, or it triggers the cell to beat erratically. So potassium, just remember that potassium is just not supposed to be high in the extracellular space. The difference between skeletal muscle and neuronal membrane potential: skeletal muscle maintains a more negative resting membrane potential. As you can see, it's around minus 90 millivolts. More negative volt. It's a bit more negative because you have an increased potassium gradient, as you could see, and there's also increased chloride gradient, and there is greater permeability for chloride, and that's why it has a bit more negative charge on the inside. The T-tubule membrane contains chloride channels as well, and that contribute to the resting membrane potential. Unlike neurons, the skeletal muscle has only one type of voltage dependent gate. I didn't mention, but the voltage gate can be active, inactive, or rested. So when the voltage gate, for example in the neurons, are activated, at the end of the potential they have to become deactivated in order for the potassium channel to be open. In the skeletal muscle, you don't really have the inactive versus deactive voltage gate. So you have different types of voltage gate. So when the voltage gate is activated, it will have to become deactive, and then it has to rest or become at a resting state before it can be reactivated again. And that's the case for the neuronal cell. With threshold voltage is reached, it quickly opens the sodium channel and you have the depolarization. So if you don't remember anything, depolarization, the sodium voltage gated channels are open and you have an influx of sodium within the cell. The sodium voltage dependent gated later closes and depolarization stops. The same voltage threshold that causes the opening of the sodium gate is the same voltage that opens the potassium gate, but they are slower. So opening about the same time that the sodium gates are closing, the potassium rush out, repolarization occur, and then you have the sodium potassium pump and larger molecule ions that return the membrane to the resting membrane potential. There's also a difference between the cardiac and the skeletal muscle physiology. I think I mentioned some of them. Action potential for skeletal muscle is caused by opening of fast sodium channel. Action potential in cardiac muscle caused by fast sodium channel as well as slow calcium channel. So just remember that calcium is also an ion, just a very important ion for action potential. So the sodium has to be released first, then the calcium, and that would initiate the contraction. But in order for the cardiac muscle to carry out the whole action potential, you need the sodium and the calcium channels. And you have different types. You have slow, fast, and L-type, different types of channels. You don't have to know them. So this is just going a little bit over the muscle contraction again. I don't even think I need to mention it because I think I covered it comprehensively before. But in terms of the resting muscle, the troponin I is bound to the actin and tropomyosin. So troponin I is bound to the actin tropomyosin. At rest, myosin head contains tightly bound ADP. At rest, there's ADP that is anchored to the myosin, and there is also troponin I that covers the region on the troponin. Following action potential, calcium binds to troponin C, and this will result in weakening of troponin I, and this will cause the exposure of the myosin binding site. This is easy to remember. Remember, you have different types of troponin: troponin I, troponin C. There's another one. C is what will bind to the calcium, CC. And eventually you'll have a cross bridge that is formed, and the ADP has to be released, because remember that the ADP is bound to the myosin head in the resting state. So the ATP will quickly become free, and this when you have the power stroke and the sarcomere is shortened. This is just showing the diagram, and I guess it goes back to rest where the ADP is basically bound to the myosin again, and the troponin I is bound again to the actin and tropomyosin. Excitation, contraction, coupling is basically a process by which depolarization of muscle fiber initiate action potential. Action potential is transmitted to all fibers via T systems. You're already familiar with the T tubule. This is a sarcolemma, nothing that you've never seen before. Only difference is now you're seeing that they're actually sodium, potassium, sodium, ATPase pump, all of these ion channels are actually there. So the action potential is transmitted, as we know, to the T system, and this is when the sodium, so the sodium here is influx into the T tubule, and it triggers the calcium from the terminal cisterns. Remember that you have the sacrotubular system, the terminal cisterns and the lateral sac, and the calcium is going to be released from the sarcoplasmic reticulum in order to bind now to the troponin C. Depolarization of T-tubule membrane activates the sarcoplasmic reticulum. It activates the sarcoplasmic reticulum via the dihydropyridine receptor. This is a calcium channel receptor. Dihydropyridine are voltage-gated calcium channel in the T-tubule membrane, and the ryanodine receptor, they're also calcium channel receptor. They're active also in the cardiac muscle, and then trigger the influx of calcium. The difference between the dihydropyridine and the ryanodine receptor, one is voltage gated and one is ligand gated. Why ryanodine is ligand gated? Because calcium is a natural ligand. Remember that the ligand gated channel respond to a ligand. Ligand ion can be a molecule, as we mentioned. It can be an ion, in this case calcium. It can be a neurotransmitter. It can be a drug. Just giving the detail of what the steps of the muscle contraction is. And this should be showing smooth muscle contraction. There's a slight difference with smooth muscle contraction, where the extracellular calcium is basically what the muscle will use. Calcium will bind to a thing called calmodulin and will create the whole actin, myosin, and contraction from the extracellular space. So the calcium from the extracellular space, and the mechanism should be the same. The membrane potential should be around, resting potential should be around 50-60 millivolts. So this is the last one. I'll try to go through it as quickly as possible. This is the cardiac excitation. The structure that make up the cardiac conduction system are the SA node, the sinoatrial node, the internodal atrial pathway, the AV or atrioventricular node. The bundle of is the Purkinje system. I think you've seen the Purkinje fibers. And remember that we said that the SA node, well, we didn't say that the SA node is the cardiac pacemaker, but these are the natural pacemakers in the heart. They are just spontaneously firing. So if you look at the diagram, you realize that the SA node is in the right atrium. It's below, somewhat at the interface between the superior vena cava. This is a big vein that is taking blood back from the head. And the right atrium will have the sinoatrial node, which is a natural pacemaker. This pacemaker contains cells that will fire 1 per minute. So this is why I have a heartbeat of around 60-80 beats per minute. It's because of the sinoatrial node being a natural pacemaker. In terms of the internodal pathway, the SA node will be connected to these pathway, and of course, yeah, the AV node will be connected to the SA node, and you will also have the atrioventricular node. These are the ventricles down here. This is the atrium, right? Atrium, left ventricle, right ventricle, and you have an interventricular septum. So you will have atrioventricular node that is basically between the atrioventricular septum, and you'll also have a division of the atrioventricular bundles or the fibers, and they'll divide into a left and a right and give you a bundle of His, left and a right. So the left one will basically go down to become Purkinje fibers. Remember, we spoke about Purkinje fibers. I think I showed you one on histology, and this is basically the mechanism by which the heart generates its own electrical activity. The SA node is the primary pacemaker, but the rest, I think, can generate their own electrical activity. Might not be the 60-80 beats per minute. This is just histology showing this supposed to be SA node, just supposed to be showing, just SA node. It might look confusing. I wouldn't even ask you to find it on a histology slide, because you more than likely won't even be able to see it unless we have very good slides or cuts. This is the bundle of His, and this should be showing the Purkinje fibers. The difference between these, the other part of the cardiac cell, is that the striations are less, and you have less mitochondria. You have less stuff there, but you would actively have to pay attention to it to realize. Like, for example, this is the ventricle with the muscle, more muscle than anything else, so you'll have more striations. But if you compare it to this area, realize that there is just less to observe in terms of muscular structure. This would be the Purkinje fiber. And I already told you all of this, the location of the SA node in the right atria, and the impulse will pass from the SA node through the atrial pathway to the AV node, to the bundle of His down here, then to the Purkinje system. So it's just basically where the impulses will be transmitted. So the pacemaker is here in the SA node, and the impulses will just be generated to the atrioventricular node down to the right, as well as the left bundle branch. And to the Purkinje. In terms of the electrical properties, the resting membrane potential is around negative 80 millivolts, and the action potential vary. So naturally, remember that you have the regular cardiac myocytes that will have their natural action potential. Then you remember that the pacemaker cells will have a different mechanism. The difference in the cardiac muscle is that when you have depolarization, you have a plateau. So it's like how the sodium just shoot up and then it just fall. You'll have a plateau for the cardiac depolarization before you have repolarization. And depolarization can last 2 milliseconds. Plateau and repolarization can last 200 milliseconds. Because remember, the plateau is like a pause in the whole thing. If you look here, this would be where the plateau is. So in terms of the action potential of the cardiac muscle, as in other excitable tissues, changes in the external potassium concentration that affects the resting membrane potential. The change in the external sodium concentration will determine the magnitude. So in initial depolarization, it's basically the same. You have the opening of sodium voltage gated channel. That's basically what you have in the nerve. So in the skeletal muscle, the cardiac muscle, it's the same. You have the sodium channels that are open, and then you have the calcium channels that are open. You have the sodium channels that are open quickly, then the sodium channel closes, and then you have the fast calcium channels that are open, and the potassium channels are closed. The phase 0 is the sodium channels opening. Phase 1 is rapid repolarization. It's due to closure. So the 1 now is when the sodium channels are closed, and then you have the opening of the potassium channel. Phase 2 is basically when you have a plateau, and it's due to slower but prolonged opening of the voltage gated calcium channel. So the plateau is as a result of the slow calcium channel. Phase 3 is the final repolarization to the resting potential, and it's due to slow closure of calcium, opening of the potassium. Phase 4 is back to the resting membrane potential, and there is a delay in increase of potassium. So this image is basically just showing the resting membrane potential around 80. Depolarization, as we said, is a rapid influx of sodium, and after that rapid influx of sodium, the sodium gate is closed and it opens the potassium gate. When it opens the potassium gate, it will basically cause an efflux of the potassium, and then you'll have the calcium channel that opens. The calcium channel is what creates this plateau, low calcium channel. And then you have a repolarization and back to the resting membrane potential. Cardiac myocyte contain 2 types of calcium channel. You have the T and L type, but the calcium current is mostly due to the slower type of calcium. And I guess that's why you have the plateau stage. This is just showing the conduction velocity from the atria, the AV node, the his Purkinje, and the ventricle. In terms of the pacemaker potential, there is the gap junctions, as you already know, between the cells that will help to propagate the rapid conduction. This is just showing the action potential in the SA node. You have the inflow of sodium, inflow of calcium and sodium, the outflow of potassium, pacemaker cells, because they're natural pacemaker. Go through the details so much of that, it's not so relevant. It's more of just understanding action potential in these nerves, muscle, because that's the bigger part of mechanic, how actions are carried out, how things move, what is needed to create the action. So yeah, that's it.
So Miss, would you want us to study the signal transduction pathways that lead to the excitation, like the one for smooth muscle?
If I should test you, I definitely wouldn't test you so much on smooth muscle. You already understand the bigger picture as it relates to muscles in general and contraction. So I would say focus more on, I highlight some stuff in red that would be nice for you guys to know. Well, this obviously you don't need to know that, but in terms of know that they have different ion channels, I would say know the difference in terms of the intracellular and the extracellular compartment of ion, because those are what's important to create the action potential. I would say know the different types of ion channel: ligand, mechanically gated, voltage gated. This is nothing that I can ask a direct question on. But in terms of the action potential, I would say no. On average, yes, the resting membrane potential, but the resting membrane potential can vary. So even if I say minus 70 for nerve, minus 80, minus 90, blah, 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 it can vary or fluctuate depending on equilibrium potential. So that is not even something that is highly specific. For action potential, though, I would say just know that naturally the threshold, depolarization, repolarization, what do you expect? And just know that the resting membrane potential, there is inactivity, there is nothing happening in the cell. Yeah, it's important to know that potassium and sodium differ, as I said, and the sodium-potassium ATPase pump is important for regulating the sodium-potassium within the cell. Yeah, this is just showing the same thing: depolarization, repolarization threshold. You can know nodes of Ranvier. That nodes of Ranvier have a lot of potassium and sodium voltage gated channel, and these are just between the myelin sheath that helps to propagate rapid action potential. Cardiac and smooth muscular pacemaker cells, that's important, that they can generate some form of activity intrinsically or automatically. This you guys should already know. See if there's anything I probably would want you to know these. And yeah, that's it.
Thank you.
Any questions? Any other questions?
Regarding the quiz for next week, it's the first 5 lectures, right?
Yes, first 5 lectures. So if you guys have no more questions, then can I say goodbye now?
Okay,
Yes,
so see you next week.
Okay, no problem. Thank you.
Bye.
Bye.